Okay, so I'm going to talk about some of the labs that we've done over the last semester. Specifically, we're going to start over here with the pentose phosphate pathway. First, we're going to talk about ribose and CO2. So ribose is a sugar that is synthesized from glucose, obviously via the pentose phosphate pathway. So first we have glucose 6-phosphate. That is converted to ribulose 5-phosphate through a series of reactions catalyzed by enzymes such as glucose 6-phosphate dehydrogenase and 6-phosphogluconate dehydrogenase during the oxidative phase. This pathway also produces a byproduct, as we can see here, of CO2, and we have 2 NADPH. These will serve as important electron transporting molecules to be used in subsequent pathways that we'll go over. The product ribulose 5-phosphate can be converted into different molecules. One of these is ribose 5-phosphate that we have right here. So that is an important sugar that will be used in the synthesis of DNA and RNA building blocks because ribose is a building block for the beginning of the synthesis of nucleotides. Which brings us over here to nucleotide synthesis. So the ribose created from the pentose phosphate pathway that we have right here is the building block for IMP, which will then be converted to AMP. So ionosine monophosphate, or IMP, as I said, acts as the, or helps with the synthesis to become AMP. AMP then catalyzes the addition of aspartate to IMP to produce adenylosuccinate, this is then lysed and produces AMP and fumarate. AMP is then converted to ATP via the enzyme kinase and the electron transport chain. This ATP will be used to synthesize DNA. ATP is used as the energy substrate throughout biochemical pathways that we have reviewed over this course, such as glycolysis, glyconeogenesis, and the citric acid cycle, which we're going to move over to glycolysis here. Some of the enzymes that are used to convert glucose to pyruvate, which is our end product down here. So we start with glucose 6-phosphate that we got over from our first page. And that becomes fructose 6-phosphate via phosphoglucose isomerase. And I won't bore you with the entire cycle, but as you can see here, we do put ATP in, get ADP out. So as you can see, as we go along here, our end product is then pyruvate. This brings us to the citric acid cycle. Pyruvate enters the citric acid cycle, which is a cyclical series of reactions whose main function creates byproducts such as NADH and FADH2, as we can see right here. So these NADH and FADH2 are very significant electron carriers that will transport electrons into the transport chain, which brings us to page 11 that we have here, as well as up here, ATP synthesis. So byproducts from glycolysis and the citric acid cycle are utilized to help transport electrons to the electron transport chain within the mitochondrial matrix. Basically, the whole point of the electron transport chain is to create ATP via ATP synthase. The electrons are transported down the electron transport chain, and you can see the series of reactions here catalyzed by the proton gradient across the mitochondrial membrane. The same gradient is utilized by the enzyme ATP synthase. This converts ADP into our building block energy compound, which of course is ATP, and we have H2O or water as a byproduct of this reaction. During the electron transport chain, protons move down their concentration gradient and into the matrix. This process catalyzes the conversion of ADP to ATP via oxidative phosphorylation. So this is a little bit about how we get ATP from CO2 and ribose.